So my name is Ilya Novikov. I'm a member of the LICHTARGE computational biology. I'm a member of the LICHTARGE computational biology group from Baylor College of Medicine, Houston, Texas. And essentially today I'm going to be talking to you about a couple of ways I developed to predict protein Go terms. That's all this fancy title means. Okay, so why do I care about predicting Go terms? Well, the gene ontology suffers from the same problem as the rest of bioinformatics, is that you have way too many sequences and not enough functional annotations. This, for example, is exemplified by human annotations in UniProt, where you can see that the total number of annotations is going up rapidly, but the average number of annotations each protein has is severely lagging behind. So, and that, of course, is because roughly 30% of the best annotated proteins account for roughly 70% of all annotations. So you end up with this whole cohort of proteins which are severely under-annotated. So, and we would like to try to predict Go terms for them. So, so thankfully, my mentor in our lab is very well versed in the field of functional inference. We have done it in many different contexts using different approaches. Some of them listed here. Now, all of these approaches had the same model, the same pipeline, the same general pipeline, is that we had some source of information, be it PDB structures or literature or sequences. We tried to extract knowledge from the data. We then tried to represent that knowledge in some sort of a mathematical framework, which could then be used to reason upon and predict novel discoveries. So I wanted to apply the same general pipeline to the gene ontology and the prediction of gene ontology terms. So I wanted to take the gene ontology, represent it in some way, and then apply reasoning to predict novel protein Go terms. So the first model I came up with is a very simple low-level model which employs non-negative matrix factorization, where for any one species, I basically take the annotation corpus for that species, which is, as many of you know, is simply a file that maps proteins to Go terms. I then convert this file into an adjacency matrix where we have terms on one axis and proteins on the other axis. And if a protein is annotated with a Go term, you put a 1 here. If it's not, you put a 0. So you end up with an adjacency matrix of 1s and 0s. So this sort of representation is amenable to reasoning by matrix completion using NNMF, where NNMF predicts novel edges based on the similarity of different proteins inside the matrix. So yes, once you apply NNMF to your original input term protein matrix, you end up with a much denser matrix that has all of these additional edges. And all of those additional edges essentially represent novel predictions. All of them have weights assigned to them, and those weights can be used to rank and evaluate all of these predictions using ROC curves. So we evaluated this model using the two standard evaluation methods in our field. One was tenfold cross-validation, where we left out iteratively 10% of our matrix and tried to predict it back using NMF. So we did this for 3 different species. It performed fairly well in all three of them. So we expanded this to quite a few more species from the CAFA challenge, and again, the mean AUC for the cross-validation was quite good. So next we tried the classical timestamp approach, where we basically went to the archived version of Go from July 2016. We made predictions based on July 2016 data, and then we compared our predictions to what was actually known in the gene ontology by January 2017. So this was essentially a simulated CAFA challenge. So the difference between these two matrices is 6 months. So again, in our 3 model species, the timestamp validation seemed to perform quite well. We did it for 19 species from the CAFA challenge, and yeah, so mean AUC was close to 0.9. So that was quite encouraging. So once we did these 2 basic validations, we asked a couple of follow-up questions. So the first question I was quite curious about is whether having all 3 ontologies, biological process, cell component, and molecular function, all in the same matrix benefits prediction or not. So I decomposed my original huge matrix into its 3 components, CC, BP, and MF, and I did cross-validation on all of them individually, and I found that yes, using entire Go as your input data is slightly better than trying to do predictions on individual matrices. So the second follow-up question I had was regarding data availability is, does having more data improve prediction? So to answer this question, I did retrospective prediction starting all the way back in 2012. And so I did retrospective prediction for each of these years. And I found that as you move further to present date, and as more and more annotations you have, the better your retrospective prediction is. So having more data certainly seems to benefit this this model. So and which gives me hope that next year it will even be better and so on. So okay, sorry. So the take-home message for this first part is that this low-level representation and reasoning model is quite robust. It does well in retrospective validation. It does well in cross-validation. It seems to work best with when all three ontologies are combined together, and it seems to improve as more data becomes available. So, so we use this model to make predictions for the CAFA3 challenge, and I'm certainly looking forward to looking at the result tomorrow. So, okay, so this model was okay, but it was somewhat unsatisfying because it was simply a very low-level representation of the term protein matrix, and I wanted to do something more fancy. So one of my lab mates suggested, well, why don't you try to make an actual bona fide protein-protein network which is based on the gene ontology. So, and that's exactly what I did. So I came up with a method, so I came up with a model at the center of which you have this protein-protein GO-term similarity network where the edges between proteins reflect the similarity of those two proteins based on their GO-terms. So how do we calculate GO-term similarity between a pair of proteins? So you can do this using a very well-established method called Resnick semantic similarity, where you basically have two proteins, 1 and 2. You compare the GO-terms belonging to these proteins in a pairwise fashion, and then and evaluate how similar the individual GO-terms are to each other. So for that you use Resnick similarity. Basically, Resnick similarity looks at the most informative common ancestor of two GO-terms, and the deeper and the more informative, the more specific the ancestor is, the more similar the two GO-terms are. So once you establish pairwise similarity between every possible pair of GO-terms belonging to these two proteins, you can then essentially average these scores to get a final similarity score for the two proteins. So now that we know how to calculate GO-term similarity for two proteins, we can do a pairwise comparison for all proteins within a species to come up with a protein-protein similarity matrix. And then to convert it into a network, you simply drop out all of the least similar edges from it. So this way you end up with a protein-protein network. So to reason on it, you can use global information diffusion, a method we've employed in our lab on numerous occasions. The way it works is if you have some label information, for example, that these two proteins, for example, have the motility GO-term, so you label them in the network, and then you diffuse this label information to all of the other unlabeled nodes. So, and once the diffusion is done, all of these previously unlabeled nodes will have some amount of label information, which reflects how likely this node is to have the same label. So, and again, you get a z-score out of it, and you can evaluate it using ROC curves. So, so we tested this approach in one model organism. We constructed a biological process network for pputida. We found that it performs well in cross-validation. Again, the principle here is the same. You label all of your GO-terms in the networks, then you leave some of them out, and you try to predict them back, and it seems to work fairly well. Then we also did this in a retrospective study, where we built a biological process network for this species using its annotations from July 2016. And then using diffusion, we tried predicting goal terms which were discovered by January 2017. Now it's, and it's certainly, it's not great, but it's certainly above non-random. So it seems to work well here too. So, okay, so it's like, I've been speaking too fast, I guess, because that's all the data I have. Okay, so the take home from these two very simple stories I showed you is that we have two methods for predicting protein Go terms. Both of these methods seem to do well in cross-validation. Both of them seem to do well in retrospective prediction. One of them, NMF, benefits from using all of the available data and not separating it into submatrices. And it also seems to benefit from having as many annotations as you can get your hands on. So, future directions, we would like to see if we can combine these two approaches, which is actually not particularly hard. For example, you can take your protein-protein network and then use NNMF to predict novel edges in it, and then use it for label diffusion. Or you can, for example, take your annotation corpus, use NNMF to predict novel labels, and then use those labels in your protein-protein network for diffusion. So you can basically sequence these two, you can basically stack these two methods back to back. And I'm very curious to see if that improves performance. Also, since gene ontology is so universal, and many non-protein entities also have Go terms, stuff like diseases and drugs, I would like to see if I can make a combined protein drug disease network or matrix as well, and see if we can predict disease and drug associations using this same approach. Okay, so acknowledgements. A big thanks to the LICHTARGE lab, and especially Angela Wilkins, who came up with the suggestion of trying to create protein-protein networks based on Go term similarity. Big thanks to Huiyan Zhang, who suggested trying NNMF, as well as to Sam Regenbogen, who is our local resident NNMF expert. And of course, thanks to Olivier LICHTARGE, our mentor. He's hiding right here. All right, and thanks to the funding agencies, including the NSF, for providing the travel grant. And if you want to talk to me personally, you can do it tomorrow between 1 and 2 p.m. at poster B158. So, thank you.
Thanks for the talk. So I have a question about the first part of your talk with NMF. So in my experience, NMF is like a very bad technique if your input matrix has like 0 rows or 0 columns. So how can you actually do a protein-centric or Go term-centric validation with this method?
I don't quite understand your question.
So,
I mean, you have a matrix of Go terms against proteins.
Yeah, so if, and you learn like these low-dimensional factors, right?
Yeah.
Yeah. So how can you, let's say, find predictions for a new protein? Let's say I give you a new protein.
Okay, great. Okay, so great question. So this question is very readily exposes the weakness of both of these methods. Basically, for any of these methods to work, you have to have some pre-existing Go terms already annotated to a protein. So if your protein is very sparsely annotated, you're probably not going to make any predictions for it. So
yeah, that's what I said. Like, so basically, if you have like a 0, like you have the whole column in a matrix is either sparse or it's 0,
right,
then
right.
you cannot predict anything with this.
It won't work. So
yeah. Okay. All right.
So my apologies, I'm not a magician.
But can you handle this problem with the other method, with the network?
With the network. No, not really, because if you have 2 proteins which only have a few Go terms, those Go terms are going to be very high-level Go terms, not very specific, and as a result, this edge between these 2 proteins is going to have a very low similarity score. So when you threshold your similarity matrix to make your network, the edge between these 2 proteins disappears, and if a protein is very poorly annotated, this node will end up with having no edges. And because it has no edges, it will be essentially dropped out of the network. So.
Okay.
So yes, this method also unfortunately only works when you have a sufficient number of Go annotations.
All right. Thank you.
So just to point out, so my original goal for this So I listed a bunch of different approaches we had in our lab for data modeling and prediction. The salient point here is that I've been trying to use gene ontology as the input source. So yes, if you have no annotations, then you have no data and you can't really represent it to reason on it. So.
But it's not really that you have no data. I think you have a sequence. So I guess my question is, you're ignoring, it seems like, I might have missed it, but it seems like you're ignoring the kind of the basic thing that we have here. And also, I guess I could point out the lack of that baseline, what BLAST would do. I think that would be important. But I've seen many of such methods in drug target and many other predictions. People just feed it into a matrix factorization or other things. But somehow it ignores some of the biology. And I wonder, for example, why would that be able to recover sequence in the first place? How could we ignore it? Right? You're expecting by using a method like this that it can do without sequence, do well.
Well, I mean, this method doesn't take sequence into account at all. So
I understand, but why would it perform well? How does it recover sequence, right? That's the.
It doesn't recover sequence.
So we think that sequence information contains information about function, not just sequence, there's systems data and other types of things. But I think just by putting that into a matrix, it suggests that that data is possible to recover.
We will have to talk about this one-on-one because I'm not quite sure how to recover sequence information from.
I'm not saying you should recover it, but the information in the sequence that recovers that provides Go terms or function, somehow you're bypassing it, and I'm not sure where that link is. How you're doing that?
I see.
Or how you would expect to do that?
I see. Okay.
Yeah, I had the same question, but is it possible you're applying this on IEA annotations, and therefore maybe the original method is using the sequence and then you're doing matrix completion?
Yeah, correct. So we include automatic annotations.
Okay, so this might be particularly good for post-processing. And
oh, yeah, good point.
Implicitly using sequence annotation because you're incorporating IEA.
Yep. Yeah, correct. Good point.
Okay, now I understand.
Yes, I guess I didn't think about this way, but thank you.